Hello students, we have just finished the first chapter under genetics, that is principles of inheritance. Continuing with the second chapter in genetics, equally important chapter, the molecular basis of inheritance. This chapter, molecular basis of inheritance, will be very much important both for NEET as well as your board exam, right? Today I will introduce DNA and then we shall proceed. Only introduction part, this one is there. This particular session is only for introducing the chapter. We will go into the main chapter from the next session itself. All right, so this chapter, molecular basis of inheritance. First, to introduce with, we look at DNA. DNA or deoxyribonucleic acid. This DNA is a long polymer of deoxyribonucleotides, right? So here DNA is a polynucleotide. DNA is a polynucleotide. It is a long polymer of individual units called deoxyribonucleotides. Next. The length of the DNA, is usually defined by the number of nucleotides or base pairs in it, right? So length of the DNA you are actually defining with the number of the nucleotides or the base pairs present in it, and that will be the characteristic of the organism. So that organism's length, the number of base pairs, will be its characteristic, right? Now we shall see certain examples of that length of DNA. If you see the length of DNA, if you see here organism, then what will be its genetic material? And here we shall see the number of nucleotides or base pairs present in it. First, if you see this virus, in this case of this virus, the genetic material is a single-stranded DNA, and it is linear. You will find that it has 5,386 nucleotides. Second one, lambda virus. Lambda virus, it is double-stranded DNA it has, and it also has a linear DNA, and it has 48,502 base pairs present in it. Then you have Escherichia coli. Escherichia coli, it has double-stranded DNA which is circular, and it has 4.6 into 10 to the power of 6 base pairs. And you have humans have double-stranded DNA, and it is a linear DNA you have, and 3.2 into 10 to the power of 9 base pair, it is the haploid content. So these 4 data are actually given in NCERT, so you should remember these 4 only. So this is what we have discussed: DNA, deoxyribonucleic acid, long polymer of deoxyribonucleotides. The length of the DNA defined in terms of number of nucleotides or base pairs present in it, and that will be the characteristic of that organism. And these 4 organisms, their genetic material and the number of nucleotides you have to remember. Next. Now if you see the chemical structure of DNA, right, or else chemical structure of deoxyribonucleotide, if you see the structure of DNA polynucleotide, you will find that the basic unit of DNA is a nucleotide, right? The basic unit in DNA is a nucleotide, and this nucleotide is made up of 3 components. And what are those 3 components? A nitrogenous base, a pentose sugar, and in case of DNA it will be deoxyribose, and a phosphate group, right? So here what I am saying is DNA is a polynucleotide. Its basic unit is a nucleotide, and nucleotide is made up of these 3 components: 1, a nitrogenous base; 2nd, a pentose sugar which is deoxyribose type; and 3rd one, a phosphate group. Next. Nitrogenous bases are of 2 types, right? Nitrogenous bases are of 2 types. One you have purines. One type you call it as purine. One type you call it as purine, and the other type you call it as pyrimidine. So 2 types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine and thymine and uracil. ठीक है? I am saying nitrogenous bases. Nitrogenous bases two types, purines and pyrimidines. Purines, adenine and guanine. Remember these, the purines, adenine and guanine are common to both DNA and RNA, right? So here the purines, adenine and guanine, they are common to both DNA and RNA, but cytosine, this pyrimidine cytosine, the pyrimidine cytosine is common to both DNA and RNA, right? Though purines are common, adenine, guanine common to both DNA and RNA, but only cytosine, the pyrimidine cytosine is common to DNA and RNA, because in DNA you have the, in addition to cytosine you have thymine, right? So you have thymine in addition to cytosine, but in RNA instead of thymine you have uracil, right? So here pyrimidine, the common pyrimidine to both DNA and RNA is cytosine. DNA will have in addition to that thymine, RNA will have in place of thymine it has uracil. So that is about nitrogenous bases. Now so nitrogenous bases are of two types we have seen. Also pentose sugar is a component of a nucleotide, we have said, right? Pentose sugar, you have two types, deoxyribose and ribose. Deoxyribose pentose sugar is found in DNA, and ribose pentose sugar you found in RNA, right? Now you'll find that the pentose sugar is attached to, the pentose sugar is attached to the nitrogenous base by an N-glycosidic linkage. The pentose sugar is attached to the nitrogenous base by an N-glycosidic linkage to form a nucleoside, right? So nucleoside, simply speaking, nucleoside is equal to, in this case, deoxyribose sugar plus nitrogenous base, right? So simply nucleoside is equal to sugar plus nitrogenous base, right? And these nucleosides, you can say, in case of RNA it is adenosine, in case of DNA you call it as deoxyadenosine. Similarly, guanosine or deoxyguanosine. Similarly, cytidine or deoxycytidine. Then you have deoxythymidine. So these all are nucleosides. Remember here, what is a nucleoside? Nucleoside is sugar plus nitrogenous base. In case of this, this is what I am showing here is deoxyribonucleoside. So here the nucleoside in DNA is deoxyribose sugar plus a nitrogenous base. So pentose sugar is attached to the nitrogenous base through this particular bond, N-glycosidic linkage. So you have now got a nucleoside. Then so you got nucleoside. Now the phosphate group is attached to the fifth hydroxyl group of the nucleoside to form the nucleotide by phosphoester linkage or phosphoester bond, right? So here nucleoside which is there, this nucleoside, this is joining the phosphate group, and you are now getting the nucleotide, right? And here this bond is phosphoester bond. So the bond between the nucleoside and the phosphate is phosphoester bond, right? Now, so now you have got a nucleotides. Two nucleotides. You have got here first nucleoside, then nucleotide. Two nucleotides can be joined in this manner by phosphodiester bond to get a dinucleotide, right? So when two nucleotides are joined, what you will get is dinucleotide, and this bond you call it as 3-5- phosphodiester bond, right? Now more and more nucleotides will join to form a polynucleotide chain, right? So what we have seen is how a nucleoside is formed, and how the nucleotides are joined to form a polynucleotide chain. Remember what are the bonds involved, what are the linkages involved, and they are very important because the different types of linkages are very important. So here we have got a polynucleotide chain. Now we'll see how these bonds look like actually. How the bonds look like. So what I have said here, two nucleotides are joined, and these two nucleotides are linked to 3-5- phosphodiester linkages. First you have got one nucleotide, another nucleotide you have got, two nucleotides are joined by phosphodiester linkage, right? And such nucleotides are joined to form a polynucleotide chain, right? Now we shall see the structure of a polynucleotide chain. See, actually this is the deoxyribose sugar. Here you will have the oxygen atom like this, and how it is formed, see. Here this will be, in this case of pentose sugar, carbon atom 1, carbon atom 2, carbon atom 3, 4, 5 and 6. Sorry. 2, carbon atom 2, carbon atom 3, carbon atom 4 and carbon atom, this is 5. Similarly, this is 1, 2, 3, 4 and you will have here fifth carbon like this. Same case here also, 1, 2, 3, 4 and here you will have the fifth carbon. So in this case, so one particular chain, one particular DNA strand, this is one DNA strand, this is another DNA strand. So one chain, one polynucleotide chain, this is one nucleotide, this thing from here to here, this is one nucleotide. One nucleotide contains one sugar and one phosphate. So one nucleotide here it is containing one pentose sugar, here one phosphate, and here you will find that there will be present the nitrogenous base. So one particular nucleotide, phosphate group, pentose sugar, and this particular nitrogenous base. So this is one nucleotide. Similarly, this one phosphate group, pentose sugar, and this nitrogenous base. This is another nucleotide. This is another nucleotide. So this is a polynucleotide chain. One nucleotide, another nucleotide, another nucleotide. So this nucleotide is attached to this nucleotide by this bond, phosphodiester bond. This is this bond plus this bond. It is called as phosphodiester bond. Right? So in a polynucleotide chain, you have one particular nucleotide is attached to another polynucleotide by means of this bond called as phosphodiester bond. 3-5- phosphodiester bond. This bond is called as 3-5- phosphodiester bond. So in one strand, in one particular polynucleotide chain, you will find that one end of the polynucleotide chain has phosphate group at the 5' position. This is the fifth position of the pentose sugar. This is the fifth carbon position. At the fifth carbon position of the of this particular pentose sugar, you will find the phosphate group. And this particular group, this particular end is called as 5' end of the polynucleotide chain. Right? So this particular end of the polynucleotide chain, when the phosphate group is attached to the fifth carbon of the pentose sugar in a polynucleotide chain, that is called as the 5' end of the polynucleotide chain. On the other end, on the opposite end, you will find that the third carbon of this pentose sugar of the nucleotide, the third carbon of the pentose sugar of the nucleotide has a free hydroxyl group. The third carbon of this particular sugar of this polynucleotide has free hydroxyl group. This end is called as the 3' end of the polynucleotide chain. So you have two ends. One end is called as the 5' end of the polynucleotide chain, and the other end is called as the 3' end of the polynucleotide chain. So this is one particular polynucleotide chain. In this particular polynucleotide chain, you have several nucleotides, and each nucleotide will have a pentose sugar, and this pentose sugar is attached to the phosphate group by means of this phosphoester bond. So pentose sugar is attached to the phosphate group by means of phosphoester bond, and the pentose sugar is also attached to the nitrogenous base, and this bond is called as N-glycosidic bond. So this is the bond N-glycosidic bond. The pentose sugar is attached to the nitrogenous base by this bond called as N-glycosidic bond, and the pentose sugar is attached to the phosphate group by this bond, and this bond is called as phosphoester bond, right? and one nucleotide is attached with the other nucleotide by means of the phosphodiester bond. And this entire chain, one end of the chain is called as a 5' end of the polynucleotide chain. It will have a free phosphate group. It will have a free phosphate group, and the other end will have free hydroxyl group at the 3' end. That is called as the 3' end of the sugar. Now, after that, if this is one strand, the second strand will have opposite polarity. If this strand is 5' to 3' end, this strand will be having opposite polarity. At this particular strand, in this particular strand, this particular nucleotide, if it is there, this end is 3' end. So this hydroxyl group is here. This is the 3' end here, and on the other side, you will have the 5' end of the polynucleotide chain. So here, the DNA strand has opposite polarity. The DNA strand has opposite polarity because if one end of the strand is between, if one polynucleotide chain runs between 5' end to 3' end, if one polynucleotide chain runs between 5' end to 3' end, other polynucleotide chain will run from 3' end to 5' end. So we say here the two chains are anti-parallel. So the two chains are anti-parallel in polarity, right? So here, this is 3' end, that is 5' end, anti-parallel polarity. And remember, the backbone of this polynucleotide chain, this backbone of the polynucleotide chain, the backbone of the polynucleotide chain is made up of sugar and phosphate, right? The backbone of the polynucleotide chain is made up of sugar and phosphate. This is sugar, this is phosphate. Sugar, phosphate, sugar, phosphate. So backbone, this is an important point here. The backbone of the polynucleotide chain is made up of sugar and phosphate. That is, it is made up of, you can also say, phosphodiester bonds, right? Second, you will find that these are the nitrogenous bases. The nitrogenous bases attached to the sugar project inside. So these are nitrogenous bases which are attached to the sugar, they will project inside, right? In addition to this, remember, so this is one particular thing. After that, you will also find that in 1953, Watson and Crick. In 1953, Watson and Crick, based on X-ray diffraction data of Wilkins and Franklin, they actually proposed the double helix model of DNA. So it was Watson and Crick in 1953. So Watson and Crick in 1953, based on this X-ray diffraction data given by Wilkins and Franklin, they have given this double helix model of DNA. So what was the main point? The hallmark of Watson and Crick's model was base pairing between the nitrogenous bases, right? So one of the hallmark, one of the main point there, one of the hallmark, one of the main point of their proposition, one of the main point of their proposition was base pairing, base pairing which was taking place between the nitrogenous bases, right? These, along with the observations of Erwin Chargaff, there was this person called Erwin Chargaff. This person called Erwin Chargaff, he observed that in a double-stranded DNA, Erwin Chargaff observed that in a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine is constant and equal. So that was the observation of Erwin Chargaff. Erwin Chargaff observed that in a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine will be equal, constant and equal. So this person called Chargaff in 1950 made observations. He made observations on the bases and other contents of DNA. Right? So this person actually, Erwin Chargaff in 1950, made some observations on the base pairing and other contents of DNA. And those observations of Erwin Chargaff, are now called as Chargaff's rules. We now call them as Chargaff's rules or Chargaff's generalizations. Those observations are now called as Chargaff's rules or Chargaff's generalizations. What are those Chargaff's rules? First he says, in a double-stranded DNA, the amount of purines will be equal to pyrimidines. That was one observation of Chargaff. The amount of purine, that is adenine plus guanine, will be equal to thymine plus cytosine. So this is one Chargaff rule. The amount of purine will be equal to the amount of pyrimidine. Second rule was that the molar amount of adenine, the molar amount of, say, purine, say in this case adenine, will be equal to molar amount of pyrimidine thymine, right? And also same case, molar amount of another purine guanine will be equal to the molar amount of cytosine. So this is the second observation of Erwin Chargaff. You can say this is the second generalization of Erwin Chargaff. Chargaff's rules we have seen. Third Chargaff rule, the sugar the sugar deoxyribose and phosphate occur in equimolar proportions, right? So this is the third Chargaff rule. Sugar and phosphate occur in equimolar proportions. Fourth one, the ratio of A plus T by G plus C is constant for a given species, right? This ratio A plus T by G plus C, this is called as the base ratio, and this base ratio is constant for a given species. For example, for human beings, this base ratio is equal to 1.52, and in case of this bacteria called E. coli, it is equal to 0.93. So this is constant for E. coli, that is constant for human being. So we have now seen Chargaff's rule, we have seen the structure of DNA. Now, these two DNA strands, see, also remember the base pairing which we are saying, adenine, the base pair, the two DNA strands are, number one, complementary to each other, right? Here, the two strands are complementary to each other. You will find that the base pairing is typical. The base pairing between the two strands is typical, complementary to each other. Always remember, purine, adenine of one strand, right? Adenine of one strand will only pair with thymine of another strand, right? So always this base pairing is typical, complementary. Adenine of one strand will always base pair with thymine of other strand, and this base pairing, you have here two hydrogen bonds. It forms two hydrogen bonds in this particular base pairing. Similarly, guanine of one strand will form bond with three hydrogen bonds with cytosine of other strand. Three hydrogen bond. So these are the base pairing. So adenine will only base pair with thymine, and guanine will only base pair with cytosine, and vice versa. So you say that the two strands in a DNA are not only antiparallel, but also they are complementary. So when the two strands are complementary, you can, when the two strands are complementary, when the sequence of bases on one strand is known, right? If you are knowing the sequence of bases, suppose this is the sequence of bases in one strand. If the sequence of bases is one strand is known, since you know that the base pairing is complementary, you know that the two strands are complementary, the opposite base pairs you can easily predict. So if the sequence of bases of one particular strand is known, the sequences of the other strand can be predicted. So here, if adenine is here, you will have here thymine. If thymine is here, you will have adenine. If cytosine is here, you will have guanine, like this. And these will form 2 bonds, 3 bonds, depending upon that. So this is one thing. So the base pairing is complementary. The two strands are complementary. So if the sequence of bases on one strand is known, the sequence on other strand can be predicted. So here remember, the base pairing is complementary. The two DNA strands are anti-parallel. They run anti-parallel, right? And later when we shall see that Watson and Crick, his model actually suggested semi-conservative DNA duplication. So his model suggested semi-conservative replication of DNA. So according to that model, if the DNA replication is semi-conservative, which is now accepted, so according to that, when two strands of DNA, when each strand of DNA separate, each parent strand will form daughter strand, right, with complementary base pairing. So when the DNA replicates semi-conservatively, the two daughter DNAs which are formed, are exact replicas of the parent DNA as well as each other. So that is about today's introductory session on molecular base of inheritance. We will go now into the real molecular basis from the next session onwards, right? Thank you.